Okay, so looking at the interactions that actually occur, this all occurs usually in the secondary lymphoid organs. So we're talking about in the spleen or in the lymph nodes. You have an antigen presenting cell, and I'm going to stick with dendritic cells because those are the main ones that play this role. The dendritic cell encounters a pathogen in the peripheral tissues, and it immediately migrates back to the nearest lymph node. That's usually called the draining lymph node. Once it reaches the draining lymph node, its job is to find one of these T cells that matches its antigen and activate it. And it should be able to do this because it's an antigen-presenting cell and a professional one, the dendritic cell. It should be able to activate both CD4 and CD8 positive T cells, depending on which class of MHC it engages. Remember, antigen-presenting cells are good at cross-presentation also, so that they can actually present exogenous antigen on an endogenous pathway, and therefore activate CD8 positive T cells and alert them to viruses that the antigen-presenting cell has encountered out in the periphery. At the same time, this antigen-presenting cell is going to start secreting a bunch of interleukin-2, IL-2, which is required for T cell activation. It is absolutely necessary that you have interleukin-2 present or you don't get full T cell activation. Once these cells get activated, they go through several rounds of mitosis and clonal expansion. And then they differentiate into effector T cells and memory T cells, both CD4 types and CD8 types. And this is one of the things that has actually changed in terms of immunology thinking compared to when I took immunology. I was taught that memory cells were the ones left over at the end of the response, like they were the survivors. But it turns out that memory cells are generated at the beginning of the immune response, in case you ever see that particular antigen again. So this is the whole basis for vaccination. You don't even have to wait for the vaccine or the pathogen to be cleared from the immune system before you start building long-term immunity to it. It occurs pretty much simultaneously with the cells that are actually supposed to go out and clear the infection. So we're going to start with the two-signal hypothesis. And this is, again, the interaction between the antigen-presenting cell and the T cell receptor. So in this diagram, what you've got going on is your antigen-presenting cell, this nice bright yellow thing. It's got MHC2 with an antigen mounted in that peptide binding groove, and we learned all about those, so you know whether if it's MHC1, it's closed on both ends, and it should be a peptide that's about 9 amino acids long, and you know excruciating levels of detail about this thing now, and you're probably sitting there going, oh yeah, I remember this stuff. Cool, good, excellent, refreshed memories. All right. On the T cell side of this, your naive T cell that is just encountering antigen for the very first time, you've got your T cell receptor, and you've got either CD4 or CD8. I'm pretty sure this one is supposed to represent CD4 because it's blue, and I think your textbook was really careful about keeping the color coding consistent. So this CD4 or CD8 molecule, either way, is going to recognize the particular type of MHC, and this generates the first signal. You get signaling through this T cell receptor. The second signal comes from these co-stimulatory interactions that come from other molecules that also engage between the antigen-presenting cell and the naive T cell. At the same time, you've got a bunch of paracrine hormones and cytokines such as IL-12, IL-2 that get secreted. You'll notice that the naive T cell already makes IL-2 and it secretes more IL-2 in response. Interleukin-2 is one of the few examples of a positive regulatory loop in the body, where a little bit of stimulation causes the production of more of that same product, which triggers the production of even more of that particular product. So signal 1 is the antigen and specific T cell receptor engagement with the T cell receptor and MHC pairing. Signal 2 is contact with co-stimulatory molecules. Contact 1, contact 2. Need two sets of signals coming into the T cell for full activation. And then there is kind of a third signal that's necessary, and that comes from cytokine signaling.
That's the IL-2.
That's the IL-2 and the other necessary or beneficial, useful ones like IL-12. So what happens is this interesting phenomenon called the immunological synapse. What happens in this system is when your antigen-presenting cell and your T cell get physically really close together and those molecules can engage with each other, it acts kind of like a zipper that sort of zips these two cells together and holds them in position with each other and actually creates this little patch where the membranes basically almost contact each other and kiss. This is known as the immunological synapse. Immunological because this is immunology class and this is between immune cells. But it's similar to the synapse formation that occurs between neurons. There's this small gap that occurs between these two cells, but mostly what you've got going on here is engagement of your MHC and T cell receptor and any co-stimulatory molecules like CD80 or CD86 that will engage with CD28. There's other options too. Some of these cell adhesion molecules, ICAM1, intracellular adhesion molecule 1, can engage with LFA1, leukocyte follicular antigen 1, I think is what that stands for. CD2, LFA3, like there's a bunch of these and there's no point in trying to memorize all of them. The main ones though that we tend to see are CD80 and 86 combining with CD28. That is usually your second stage of activation. That's usually your signal 2.
Which CDs do we need to know again?
4 and 8, and in this case 28 and 80, 86. And it's CD80 or CD86.
Can you see the supramolecular activating complex in that picture?
So, yes, you can see the supramolecular activating complex in this picture. So up at the top, you've got these two images, and one is just basically a 3D reconstruction of a T cell and an antigen presenting cell coming into contact with each other. It almost looks like one's going to eat the other. But what happens is you get this ring of concentric circles that forms. So in the middle of this, in the very center of this contact patch between the two cells, what you end up with is this ring, this little central clustering where you have a bunch of T cell receptors that all cluster into one specific spot near that point of contact between the two cells. And then that forms what's called the central supramolecular activating complex, or the CSMAC. Going out one ring layer outside of that, you'll have the peripheral activating mechanisms. So usually these are things like the ICAMs and the LFAs and some of the other co-stimulatory molecules. But you can actually see these on fluorescence microscopy if you take the pictures in the right way with confocal microscope. So in the very center, you've got your T cell receptor and CD3, because CD3 is necessary and it's part of that T cell receptor complex. It's the part CD3 is needed for signal transduction. Also in that CSMAC are CD4 or CD8 and your co-stimulatory molecules and receptors such as CD28. So basically, it's this region that's colored in red on the lower part of the diagram. That's what forms the central supramolecular complex. And then the next ring out from that, the peripheral supramolecular activating complex, the PSMAC region, includes any other adhesion molecules that may engage. But it's this clustering of T cell receptors and secondary activating signaling molecules like CD28 that really help to solidify and firm up this interaction between the antigen presenting cell and the T cell. And once you've got contact, and once you've formed this initial patch with the C-SMAC and the P-SMAC regions that actually form those central interactions, what you have is T cell receptor signaling. And initially, the signaling starts with T cell receptor engaging with antigen on MHC. And then the signaling is going to really mostly come through CD3. And so T cell activation begins with a tyrosine kinase. All of this is tyrosine kinase driven. So this goes back to stuff that we learned about in cell biology, where the receptor tyrosine kinases trans-autophosphorylated, where they did each one phosphorylated the tail of the opposite chain. That same kind of event is going to happen here. And T cell activation starts with a particular tyrosine kinase known as LCK. LCK. What CD4 and CD8 actually do, other than recognize the appropriate MHC, is they help to guide that LCK, the LCK enzyme, to the T cell receptor and MHC complex. And then LCK actually is what phosphorylates CD3 and activates it. And from there, you get a cascade of phosphorylation events. So the ITAM regions become phosphorylated. They, in turn, will phosphorylate other proteins that start your signal transduction cascade. So looking at this diagram, you've got CD4 or CD8. It's supposed to recognize the appropriate type of MHC. Since that one's blue, it's CD4, which means it'll recognize MHC2 on the opposite antigen presenting cell. If that's a match, you get LCK that's actually going to transfer a phosphate group onto these ITAM regions in the CD3 cytoplasmic tails. Once those get phosphorylated, they're in turn going to phosphorylate other things like ZAP70, which transfers phosphate groups to downstream effector molecules that create these signal transduction cascades, ultimately ending in activation of the same things that we've seen before: NF-kappa B, NF-AT, and AP1, and those were on the last exam. So these signaling pathways come back to haunt us again in this particular chapter, but they're key to activating the T cells and getting them to actually perform their effector functions. CD4 positive T cells going on to activate B cells, and CD8 positive T cells going out to kill infected host cells. And yeah, there's a lot going on in this diagram. Where we pick up from here is after ZAP70 activates, for example, phospholipase C, PLC in that diagram there, which is next to that big long bar that says LAT on it. Phospholipase C is going to actually take membrane phospholipids and cleave them into two separate signaling molecules, diacylglycerol and IP3. IP3 is going to trigger calcium release from the endoplasmic reticulum, which goes on to activate the NF-AT pathway, while the diacylglycerol is actually going to activate RAF, MEK, ERK, the whole GRB2 sauce and MAP kinase pathway that we learned about before and back in cell biology. If you need a refresher, it's still there. And the PIP2 can actually activate the NF-kappa B pathway through protein kinase C and diacylglycerol. Again, so diacylglycerol can actually activate two separate pathways. And that's actually what I had already just talked through. Oops, got ahead of myself. That happens. I liked signal transduction cascades, even though it meant having to do some memorization, because it was a pathway that was linear, it was straightforward, and it was something that I could write out pretty easily in terms of like dealing with studying and learning this material. And to this day, I still think signal transduction cascades are fascinating, even if I can't say them this morning. So basically, this one event triggers 3 separate cell signaling pathways. One that ultimately activates NF-kappa B from the diacylglycerol, one that activates NFAT through the calcium pathway released from the endoplasmic reticulum, and then the MAP kinase pathway releases the transcription factor AP1. So in the MAP kinase pathway, MAP kinase or mitogen activated protein kinases phosphorylate a sequence of proteins. Usually the generic ones are RAF, MEK, and ERK. So RAFs will transfer a phosphate group onto RAF. RAF will transfer a phosphate group onto MEK. MEK will transfer a phosphate onto ERK. And ERK will finally activate the transcription factor AP1, which can then move into the nucleus and start changing gene expressions. And it's just a few T cell receptor interactions that are needed to trigger one of these signal transduction cascades. So not many, but not just one either. One could be a mistake, right? Like in this whole complex immunology thing, just one T cell receptor matching up with an antigen, you might not want to pay attention to that. That could have been a fluke. But if you've got a number of T cell receptors all recognizing antigens, that stronger signal that you would get from that is what really drives all of this signal transduction that happens and the changes in cell behavior that occur as a result of the gene activation. And you all look brain fried this morning. That's a lot to deal with. So we'll stop there, also because it's 9:49.
A quick question. Do you want us to know the master regulator?
You really should probably know the master regulators, yes. So you have protective Th17 cells and inflammatory Th17 cells. The protective ones appear to be produced in response to transforming growth factor beta, TGF beta, and IL-6. While in inflammatory cells, you need TGF beta, IL-6, and the additional presence of IL-23 to get the inflammatory type. By itself, IL-23 is not sufficient to induce production of Th17 cells. It has to be in this combination.
I think it's kind of interesting that there are dual roles for this particular subset of cells, and realistically, if I were going to go study immunology again, I would probably want to know more about them because I'm intrigued by this difference between two cells that have very similar phenotypes but very different activities. It just kind of catches my interest.
And for inflammatory, when you say TGF, do you mean TGF beta?
Yes, TGF beta. There are two kind of subtypes of interleukin 17. There's IL-17F, F as in fungi, and IL-17A, which is like autoimmune, if that helps you keep things straight on which one does what. It's not perfect, but it's close enough. Any little memory keys are helpful. Okay. These cells also come about because of exposure to the induced T regulatory cells, or Tregs. These are similar in function to natural Tregs that originate in the thymus, but these are specifically a CD4 helper subset that arises on exposure to TGF beta without IL-6. So it kind of depends on the ingredients of the recipe that you use, which is interesting because that means we've actually got a really good ability to generate large numbers or larger numbers of any of these cell types in vitro. We can literally just bathe them with like TGF beta without IL-6 and help to induce more of these T regulatory cells so that we can kind of study what they do. There's also no IL-23. So no IL-23 or IL-6 in this particular case, and those were important for the Th17s. These are highly immunosuppressive. Their job is to tone down or tamp down or turn off immune responses. This is important because eventually you're going to clear your infection and you need the immune system to basically stop attacking your body at that point. It also means that it helps to tamp down on autoimmune issues. In fact, if you deplete healthy animals of Tregs, like through genetic manipulation or through some specific techniques that we can use in the lab, those animals then go on to have multiple autoimmune outbreaks. So these are really good at suppressing autoimmunity and promoting self-tolerance in peripheral tissues. It also kind of suggests that we're all constantly fending off autoimmune issues and we don't really know about it. So I find it kind of interesting because there appears to be an increase in autoimmunity that's been going on in the last couple decades in particular. Some of that is probably just due to better diagnostics and having a better understanding of the systems, but there's increased diagnoses of like inflammatory bowel conditions, increased instances of diabetes, including type 1 diabetes has risen, increased instances of things like lupus and multiple sclerosis, and these are all autoimmune conditions. That suggests that there's something kind of not working properly in our immune systems to sort of regulate those processes, and it may have to do with something about the way we're growing up and the world that we live in now. I suspect that it's environmental, to be honest. I'm pretty sure my body can make Treg cells because, I don't have autoimmune issues all the time. And they're not constantly new ones popping up every other week. Like, something is working, but it's not maybe working as well as it should be working. The master regulator for these cells is FOXP3, and this is one that helps to shift cells specifically into the Treg subset. This is how we identify Tregs, whether they come from the thymus or whether they are induced Tregs that happen in the lymph nodes or spleen. These cells secrete TGF beta and interleukin 10. Interleukin 10 is very, very immunosuppressive. And it inhibits antigen-presenting cells. And for that matter, other T cell subsets like your CD8 positive T cells and other CD4 positive T cells. Question.
For the antigen presenting cells, does it inhibit them by like its shape, or is it blocking them from coming into?
It's chemical. It's the production of those particular cytokines cause chemical changes within the antigen presenting cells that cause them to become less active.
Okay.
And to basically turn off.
Okay.
Yeah. Why isn't it letting me advance slides today? Okay, so we had cross-regulation where in the Th1 and Th2 subsets, we also see the same phenomenon for Th17 and Tregs. So these two cell types, once you start down one of these two differentiation pathways for Th17 cells or regulatory T cells, especially the iTregs, the induced Tregs, it inhibits them from joining the other pathway. TGF-beta was key for both of these cell types. So it starts the cells differentiating from Th1 or Th2s and into one of these other two subsets. And it looks like IL-6 is the major switch that kind of allows ROR gamma T to become the major transcription factor. This is what shifts cells into that Th17 subset. And then after that, you get a choice between protective or inflammatory phenotype based on the presence or absence of interleukin-23. We need both types of cells, but they do sort of inhibit the production of the other types of cells. So there has to be some sort of mechanism to create a balance between these two populations. The normal state should probably, like when we're not infected, should probably favor production of the inducible Tregs so that we have less autoimmune issues and inflammation on a regular basis. However, if you have an active infection where you have IL-6 available, that should hopefully stimulate a little bit more antibacterial Th17 differentiation. So the key here may be not just the presence of IL-6, but the level of IL-6, if that sort of makes sense. If you have a lot of it around, that indicates infection. If you've just got a little bit of it around, maybe that's just normal resting circumstances. And so that may help to define which pathway these cells actually end up in. But once you activate the transcription factor for one particular subtype of cells, the other particular subtype gets inhibited at the genetic level. So these cells can't just like switch back and forth. More roles for IL-6 is to form follicular T helper cells. These T helper cells stay in the follicles of the spleen or the lymph node, so they don't really patrol around bodily tissues. Because of this location, they really are primarily involved in helping support B cells in their developmental pathways and differentiation pathways, which is the topic of the next chapter, which we're not going to get to today, obviously, because I can't talk that fast. I know I talk fast enough. But it appears to be the combination of interleukin-6 and interleukin-21 that shift cells to becoming T follicular helper cells, TFHs, because we don't have enough abbreviations. When I was reading this for myself, I found myself reading the abbreviation first, so I tend to pronounce it follicular helper T cell instead of TFH. These cells produce IL-4 and IL-21, and those are important for B cell differentiation. We have seen IL-4 before. Where did we see IL-4 before? Which other T helper subset? 2. Why is it disappearing? There we go. Th2s. So Th2s and the type 2 responses were highly characterized by interleukin-4, and this really shifted things toward a B cell antibody producing immune response, a more humoral response. This was particularly good at activating eosinophils and clearing parasitic infections. So we see this show up again for this particular subtype of cells, the T follicular helper cells, which reside in those follicles and basically play very assistive roles for B cells. That's not terribly surprising that both the type 2 response and T helper follicular cells are involved in activating B cells, and they use a common cytokine. So if you're talking about T help provided to B cells, it's heavily depends on interleukin-4. They express a couple of co-stimulatory molecules that are necessary for full activation and differentiation of B cells. That would be things like CD45 or CD40 ligand and CXCR5. That's a CXC chemokine receptor or cytokine receptor, chemokine. And that's R is for receptor. So this is the CXC chemokine receptor 5. And this is, among other things, what helps to attract the T follicular helper cells into the B cell follicles, and it also helps bring in B cells from the rest of the lymph node or spleen into these B cell follicles and helps to form these follicles. Manal, you had a question.
I was just going to say, so you're saying that the follicular helper T cells, they produce the IL-4 and IL-21, and they help.
They are produced in exposure to IL-6 and IL-21, and yes, they also produce IL-4 and more IL-21. Yeah. Okay, so what do these things do in an immune response? What's really different between these T follicular helper cells and Th1 cells, aside from the types of pathogens that are involved? So let's start with the ones over here on the left-hand side instead of the ones on the right-hand side. We'll work our way that way. Okay, so in the beginning, we have a B cell that encounters a T follicular helper cell. You've got your T cell receptor, MHC class 2, that's being expressed by the B cell receptor. In the next chapter, you're going to learn how and why these cells start to express MHC class 2. But remember, B cells are one of your 3 major types of antigen-presenting cells. You have B cells, macrophages, and dendritic cells, so this is one of them. They are capable of presenting antigen, so you get antigen recognition between your B cell and your T cell. Both of these cells are antigen specific. So this is highly tailored to whatever antigen your immune system has picked up. Expression of CD40 and CD40 ligand co-stimulatory molecules that help to firm up the anchoring between the T cell and B cell, and also provide supplemental signaling that basically forms a cross-check that says, yes, this is a licensed cell, this should be getting activated. It provides a very necessary second signal. You've got another set of co-stimulatory molecules, ICOS and ICOS ligand, that are expressed between the T and B cells as well. So there's a number of co-stimulatory molecules and anchoring molecules that are going to work together at the synapse to hold these 2 cells physically together. Once you get activation of the B cell, it goes through clonal expansion, so you get a nice population of cells that come out of that. This is in response to interleukin 2, 4, 21, and some interferon gamma. All contribute to producing these particular clusters of B cells. These are called germinal centers. We'll see them again in the next chapter as well. But what this does is it really favors production ultimately of plasma cells. Plasma cells are the ones that are going to go on to produce antibody. Now, if you remember back in the beginning when we started learning about B cells, we learned a few things about B cells. They have a B cell receptor. You can see that on the surface of these cells, right? B cell receptors stuck on the surface of the cells. But check out your plasma cell. No B cell receptors. They've all disappeared. In fact, now they're shedding pure just antibody. So it looks like they stop making membrane-bound B cell receptors and shift to producing soluble antibody. What process regulated that?
Splicing.
Splicing. Very definitive. I like it. Excellent. You remember this. See, you have learned stuff this semester. Okay. So something happens that causes a difference in the splicing mechanisms from this clone of B cells to the mature plasma cells. Again, that's something that we'll look at a little bit more in the next chapter. The first antibodies that are going to be produced by these plasma cells, the earliest ones you'll see in the immune response are going to be IgM and IgD, because those are the molecules that the cells have been presenting and showing, and that's the B cell receptors that you had so far. A little bit later, and in the next chapter also, we'll learn how to make the other types of antibodies that appear later during immune infections or immune responses. Okay. So it's this particular interaction between the B cells and the T follicular helper cells that favors the production of plasma cells. In contrast, what we've got here is kind of a three-way communication going on to activate CD8 positive T cells. You've got a dendritic cell, we'll call it a dendritic cell. It looks like a dendritic cell to me because of all the little processes sticking out of it. And it's actually showing off antigen in MHC class 2 to two different, it's showing the class 2 to a T helper 1 cell subtype, and from its MHC class 1, it's also displaying that same antigen in two different ways to two different subsets of T cells. MHC1 pairs with our CD8 T cells, MHC2 pairs with our CD4 positive T helper 1 cell subset. So you've also got co-stimulatory molecules that are going back and forth between the Th1 cell and the antigen presenting cell. You've got CD8 that's actually checking in on the MHC between the antigen presenting cell and the CD8 cell. And then you've got communication also between the T cell helper cell, the CD4 positive T cell, and the CD8 positive T cell through CD40 and CD40 ligand. All of this together triggers these CD8 positive T cells to go through clonal expansion, just like the B cells did, and make cytotoxic T cells. These are the ones that go out and actually kill your target cells. They do that by releasing some chemicals like perforin and granzymes, kind of similar to the ones that are made by the innate immune cells and NK cells. Yes?
The follicle helper cells, they're CD4.
The follicular helper cells are CD4, yes. All of the TH subtypes are CD4s. And all of this goes on when you get an infection. Both arms, both the cytotoxic T helper cells and the plasma cells secreted or produced from that will produce antibodies, both of these systems tend to get activated. Now, depending on what type of pathogen you have, it may shift this response to maybe favoring this pathway, especially if you've got virally infected cells, because you need CD8 positive T cells to clear virus infections. They're one of few cell types that have the license to kill your own host cells occasionally. Plasma cells, all they're going to do is secrete antibodies. Now, they can still be very effective virus fighters, and this is partly why we talk about virus responses, antiviral responses, and both virus titers and antibody titers in response to infection, or in response to vaccination in the case of pure antibody titers. When your antibody levels against the virus remain high, so if you just got that particular vaccine, you're going to have high levels of circulating antibodies for a couple months after that. You have built-in innate protection, humoral protection against that particular virus. The antibodies can get there before the rest of your immune system can do anything else. But over time, these plasma cells die off after about 4.5 months. And that means they're going to stop producing antibodies. And the levels of antibodies in your bloodstream are going to take a nosedive and they're going to fall. So then you're relying on your memory T cells, the CD8s, the CD4s, and your memory B cells. We haven't even talked about memory cell formation yet. It's kind of cool, but it's really the heart of vaccination. There's a couple other smaller subsets of T cells that you can actually encounter. These include the Th9 cells and the Th22 cells. I am not going to ask you questions about these. You can eliminate them from your study list. Know that they exist, but beyond that, they're still relatively new and relatively small subsets of cells, and their roles are a little bit less clear. Whether they exist is a little bit less clear. We're still trying to really confirm them and shore up the evidence that these represent truly distinct subpopulations of cells and that they're not just some sort of weird Th17 subtype that we didn't know about before. So, and Th17 was an example. Early experimental evidence looks like they probably are actually real, but we don't know enough about what they do yet. Okay. The other thing is that so far when we've talked about differentiation, we've talked about these cells, they start down this path to becoming T cells and they become locked in. They really can't change after they start down that particular pathway. But there is a little bit more flexibility in the T helper lineages, in these Th1, Th2, Th17, T follicular helper subsets, those guys. So when they're young, like shortly after activation, even if they start differentiating down a pathway, if the cytokine environment around them changes enough, they can actually shift toward the other pathway, whichever one is favored by the cytokine bath that they're currently in. So an example is if you take young T helper 1 cells, they can actually produce IL-4, interleukin-4, under the right conditions if you've got enough T helper 2 polarizing conditions around, so that would be IL-4. So you can actually get T helper 1 cells to make IL-4, it's just not their default sort of state. And this fluidity is part of why it's questionable whether or not Th9s and Th22s are really a thing or not. Like, we're not totally sure yet. It's tough to kind of dissect these out, especially when we're talking about populations of a few hundred to a few thousand cells. That's small, and it makes them hard to study. Okay. So these particular subsets actually play interesting roles in disease. Like, there's actually 2 different forms of leprosy, which is wild. Like, I learned interesting things about leprosy that I didn't know before either. There's the tuberculoid leprosy, which is generally milder and survivable, and the lepromatous leprosy, where people's body parts rot off and fall off. They literally decay and break off. Yeah, it's bad. There's actually some speculation. Okay, how many of you are into Game of Thrones? Have you done House of the Dragon?
Love House of the Dragon.
Okay. There's some speculation that actually one of the characters in House of the Dragon may actually have a form of leprosy, and that's all I'm going to say about that, because if you haven't seen the series and you want to, I don't want to give spoilers. But it doesn't take a lot of thinking to figure out who. Oh yeah, you'll just know. Okay, so what we see is a difference in the cytokine responses and in the T cell responses that are different between these 2 different forms of leprosy. So in the tuberculoid version, the milder version of leprosy, what we see is more production of interleukin-2, interferon gamma, and LT alpha. These are type 1 cytokines. So you see a stronger type 1 response
this is associated with a tuberculoid outcome of leprosy, the more survivable and controllable type of infection. On the other hand, a Th2 response, we see more production of IL-4, IL-5, and IL-10, which is immunosuppressive. IL-4, which shifts things towards a humoral response because B cells, IL-4, B cells, IL-4, B cells. Have I kind of made that connection for you now? Excellent, good. That may appear on the test. We see a type 2 response in the patients that actually have the lepromatous leprosy, the more severe form. So the question is, I'm guessing you know nothing about leprosy itself, but it's actually caused by a mycobacterium infection. And a lot of the mycobacteria infections are actually intracellular pathogens. They are bacteria that get inside the host cells. So for that, to control an intracellular infection, whether it's viral or bacterial, you really need a type 1 response, if at all possible. So you want high production of interleukin-2, interferon gamma, and LT alpha, and this enables the immune system to control and kill off those cells that have been infected with those mycobacteria.
So I'm looking at the chart, and it says that Th2 cells usually protect against extracellular pathogens.
Yes, Th2 cells provide better protection against extracellular pathogens, but mycobacterium is intracellular.
Yeah, so why would they take even the Th?
Because it's still bacterial, so you still see some development of of bacteria outside the cells when it's spreading from tissue to tissue.
It's extracellular Th2, Th1's are intracellular.
We actually also see a difference in HIV progression to AIDS. So this is one that doesn't get talked about a lot today, but that's partially because we've really got very good therapies today that control and prevent infection of HIV. So it's better than it used to be. But the progression from HIV infection to full-blown AIDS, which is characterized by very low numbers of circulating T cells in your bloodstream and very high levels of virus in your bloodstream. This is also influenced by a shift from a Th1 antiviral response to a Th2 antibody humoral response. The antibody humoral response is just not sufficient to control an HIV viral infection. So it's kind of important that we get these responses right. Let's see. In Epstein-Barr virus, which causes infectious mononucleosis, and also Bell's palsy, which is an infection of the 7th facial cranial nerve. That nerve actually runs from basically the base of your skull. It exits through a little hole right in here by your ear, and it fans out from just under your cheekbone, and that nerve feeds basically 1/2 of your face. You've got one on each side. This enables you to control the 2 sides of your face independently and wink or smile with half of your mouth. So there's an interesting thing about the Epstein-Barr virus. It produces its own version of IL-10. It has a viral equivalent to IL-10, which suppresses immune responses. This virus comes prepackaged with an immunosuppressive thing to try to evade your immune system. That is an evil trick. It's actually fairly difficult to clear an Epstein-Barr virus infection. That's why mononucleosis people who get that are usually it takes a minimum of 3 weeks to really recover from that. And you're not really supposed to be around other people a lot. You're going to be too exhausted to be anyway. One of the hallmarks of that is extreme fatigue. And if it gets into your 7th facial cranial nerve, you could actually have recurrent Bell's palsy. What happens with Bell's palsy, more or less, you wake up and half your face is paralyzed. It just doesn't work. How do you figure out half your face is paralyzed? Well, when you go in the bathroom and you try to brush your teeth and you can't hold the spit in your mouth from the toothpaste, and it just dribbles down and you're like, what in the world is going on? Did I sleep funny? No, and then you realize you're not blinking on that side of your face. And then you're like, oh shit, I had a stroke. So then is usually when people call their doctors. So you go in, you get it checked out, and hopefully it's just Bell's palsy. Those infections resolve well with treatment too, and most people get full motion back. So it's usually fairly transient. I've actually had it twice. I can tell you that the worst part about it is not being able to blink. You don't realize how important blinking is until you literally cannot blink your eye. You have to manually go around doing this all day. And you have to put not drops. Drops aren't enough to keep your eye lubricated. You have to basically put Vaseline in your eyeball. This does not do good things for your ability to see or drive a car. You cannot wash your face. Do not wash your hair. Taking a shower becomes very risky because soap, it gets in your eye. You can't taste anything on that side of your tongue, which is wild. That's usually actually my first symptom is like it. I always assume that I've burned my tongue. Like both times I've had it, I was like, I must have really burned the crap out of my tongue. I don't remember doing that, but it happens to me often enough that, you know, I just assume that that's the more likely explanation. Nope. So Bell's palsy is kind of an interesting one. It's actually pretty common. 1 in about 10,000 people get it.
Is it possible to actually keep the eye closed?
Is it possible to keep your eye closed? No. You have to tape it shut at night.
Oh, really?
Yes, really.
So,
Is that
I ran around with like a pirate eye patch for a while because it was just easier.
Is that kind of like Ramsey Hunt syndrome, blah, blah, blah? Because Justin Bieber has this thing where like half of his face went numb, like he couldn't do anything with it, but they're calling it Ramsey Hunt syndrome.
That's from a different cause.
Oh, okay.
Yeah. But probably similar mechanism. And yeah, half your face is also numb. So it for some people it droops. So it looks like, for most people, it basically looks like you've been to the dentist and had some Novocaine and your face is just kind of can't feel your lip. Everything feels slightly swollen. Like if you touch it and poke it, you don't get any sensation from it except from your finger. It's weird, but it's absolutely survivable and recoverable. Okay, and that brings us to T-cell memory development. How do we get from making an activated T-cell of whatever subtype it may be, whether they're Th1 cells or Th2 cells, to producing our all-important memory cells that we really need for future protection against reinfection with that same pathogen? That's ultimately what memory cells are for. So after antigen encounter, you get about 90% of those T cells are lost, which means your memory cell pool represents something like 10% of your initial population of T cells that were used for fighting the infection. So if you think about it, I don't know how we have any T cells left. From the wholesale slaughter that went on in the thymus during thymic selection, both rounds of like beta selection, positive selection, negative selection, you get to the lymph nodes, you get more selection that occurs there. You get through an immune response finally, yay, and 90% of those die. And yet you're left with enough memory cells to successfully defend you against future infections. That is so awesome. It only takes a few, because those memory cells are going to have the unique ability to respond much faster. They've basically already been activated. They're just kind of hanging out in a sort of a quiet state. Technically, it's G0 of the cell cycle. After you've cleared an infection, these will form about 35% of the circulating T cells in your bloodstream. 35. That's actually surprisingly high. But they're constantly patrolling your body, keeping an eye out for future reinfection with that same pathogen. Go T cells. In an individual who is 70 years old, that percentage increases to about 60%. They've seen more infections. Their immune systems have fought off more things. Also, their ability to produce new T cells has declined significantly because that's a thing that happens with age too. So we see a shift. We talk about the immune system getting weaker as you get older. Well, yes and no. It doesn't exactly get weaker. It just has kind of exhausted its ability to respond to new things. Okay. So for memory cells, naive effector and memory T cells All three of these particular subsets, remember, naive cells have never seen pathogen. Effector cells are the ones that are activated in response to a current infection, go out and seek to destroy the infection. So that could be T cells of the CD4 or CD8 subtypes. Either way, once they become activated and they acquire their mature phenotype, they're referred to as effector cells. And then there's the memory cells. All three of these different lineages express different combinations of cell surface markers, and 4 in particular that are used to differentiate memory cells from the rest. CD44. This increases in response to T cell receptor signaling. So one of the earliest events in activating a T cell, one of the first things that's going to be increased on those cells is CD44. You can tell an activated new effector cell by how much CD44 it expresses. We also saw CD44 during T cell development.
I thought that was 45. I'm like, did we see that?
44, 45. 45 is important for B cells. Yeah. Like I said, there's too many of these CD fill in the blanks. You know, there's at least 273 of them that I know of. I'm not memorizing all of that. Just plain no. Yeah. That's what Wikipedia is for. Thank you very much. I will look those up. Or the table in the back of the book, you know, if you happen to have one of those handy. So CD44 during development of new thymocytes was expressed on the brand new thymocytes as they were entering the thymus, but then it was rapidly downregulated as you progressed through those double negative stages. In case you were wondering where you had seen this before. CD62 ligand is an adhesion protein that is important for homing to secondary lymphoid organs. That means this one is particularly important for T cells migrating to lymph nodes or spleen. Chemokine receptor 7, CCR7, is also important for homing to secondary lymphoid organs. There is an infection. Please head to your nearest lymph node. Thank you. And then there's CD69. CD69 is actually a C-type lectin, so it's a carbohydrate, and it prevents the cells from leaving a particular tissue. So you actually maintain a pool of memory cells in the tissue that was originally infected. This is one of the reasons that there's some interest in using intranasal vaccines for influenza and for COVID, is because that's your primary route of infection. So if we can build natural immunity and keep a pool of memory cells like where the reinfection is likely to occur, because it's not going to occur in your upper arm muscle. That is not how COVID gets into the body. It's just not. So if we can generate a more natural immune response that puts some of those memory cells in the proper tissue, that actually should improve our long-term outcomes and our ability to fight off those infections in the future. Why we don't have more intranasal vaccines, I don't know, because the vast majority of people would rather take a nasal spray than a shot. Like, this just seems stupid. But it may have something to do with stability and formulation. So a lot of the shots that we get, we have to keep them very cold to keep them from degrading. And nasal sprays are usually like, you don't want to stick icy cold fluid up your nose. So nasal sprays are usually room temperature, and they're usually much more dilute. That may be part of the issue too. I'm not an expert on the development of like nasal spray vaccines. But one of you might want to be. Please fix this for us. I'm not afraid of needles, but I would much rather get a nasal spray. Especially because I understand like how the immune system works. So I would really prefer that. And they actually do have nasal spray vaccines available for flu. They just don't use them. They're not widespread, but they do exist. And if you go in and ask for one, you probably won't get it. Like that's only available like for very young kids or the very elderly, usually. Okay. Lots of text, little print. We'll work through this. There are 4 main sets of memory T cells. TSCMs, stem cell memory T cells. These have some characteristics of true stem cells. They're capable of self-renewal, which is not like what we see for our effector T cells. They retain this ability to actually proliferate and make more copies of themselves, like what we saw back in the naive T cells. These are rare, and they are the least differentiated type of memory T cell that you actually have. There is debate about where these cells come from. So there's a couple different possibilities, but the main data and available information so far seems to indicate that they actually come from naive T cells. So basically, when a naive T cell gets activated, it can take one of 2 pathways and either become an effector cell or a memory cell. But that decision gets made all the way back there at the early stages. This is not what I was taught about how immunology memory forms. I was taught that it was the leftover cells at the end of your immune response that turned into memory cells. But that doesn't appear to be true.
Like, how would it have a memory if it has only just been activated?
It doesn't have a memory, it provides you with a memory. So that antigen has now been recognized and those cells have been activated, and so you get that clonal expansion. A little group of those cells basically break off to become memory cells. And early in this process, they are stem cell memory cells. Your next level are the central memory T cells. These are called central because they reside in your secondary lymphoid tissues. They go back to the lymph nodes or the bone marrow or the spleen. They have long lives, so they will stick around for quite a while. They still have some ability to divide and replicate, but not as much. But these are rapidly activated on second antigen exposure, or third or fourth, or however many times you catch the same cold. I've picked on flu and COVID a lot this semester, so let's pick on cold viruses. And they can differentiate into several subset types depending on the cytokine environment. So these central memory T cells actually retain the ability to go on and become any of the CD4 positive T cell subsets. They're basically kind of undifferentiated still at this stage. You've taken a generic T cell and activated it partially, but not completely. And it waits until secondary antigen exposure to finish that maturation. Effector memory T cells. These are the cells that travel back and forth between like your skin, your gut, your nasal epithelium, and those secondary lymphoid tissues. They contribute to our first line of defenses. So these are some of the ones that if we actually used nasal spray vaccines, we'd be relying on or generate some of these effector T memory cells. And they shift right back into effector functions once you get re-exposure to that same antigen. So they don't have to go back to the lymph nodes, go through all that proliferation, go through all of that cytokine bath and take that days-long process. They're already there. As soon as they see the antigen a second time from an antigen-presenting cell, they can just go, oh wow, seen that before, kill. It's a much faster process, and this is why our memory cells are so important. And then you have the resident memory T cells, the TRM cells. These are the permanent residents of your previously infected tissues. You got a bunch of them for all the shots that you got as a kid that are living in your probably upper arm and butt cheek muscles, since they like to give vaccinations in one of those too. That's because those are nice large muscle groups. Little kids don't have as much muscle, in case you've ever wondered why they give you shots there, so that the needle doesn't actually go through and hit the bone, because that is incredibly painful. Sometimes on really small children, they'll actually, or they used to actually give it in the upper thigh muscle. They did that to me when I was a kid, and they actually did hit the bone on me. I couldn't walk for like 2 or 3 days afterwards. My parents didn't quite understand what was happening, but they knew that it hurt. Like I didn't make any fuss during the actual shot, but like the next day I physically could not walk. And they realized something had happened. So they asked the doctor, and they were like, yeah, the needle probably scratched the bone. That's lovely. How about no? So, how and when do these memory cells come about? Like I said, the thinking on this has actually changed from when I took immunology, which is fascinating, but we've got better information at this point. We think that these actually arise very early in the immune response. So, like I said, when these cells initially, when your activated T cell in the lymph node goes through clonal expansion, a little group of those cells is going to break off and basically become memory cells. And then we actually think that there's a progression in terms of the phenotype of these cells. So you start off with TSCMs, the stem cell memory T cells. These they mature into TCMs, the central memory T cells. But it's not entirely clear. So there's also some evidence that they can come from effector memory cells, which is what we normally think of the central memory cells as developing into. So there's a little bit of questionability about which order these two processes go in, or whether there actually is an order. It may just be a mix of both. But to the best of our current understanding, this is kind of what we're dealing with. Naive T cell encounters an antigen-presenting cell that has the correct type of antigen. This goes through clonal expansion. The bulk of these cells, 90% of them, go on to become activated effector cells, either CD4 positive or CD8 positive. A small group, the other 10% or so, develop into stem cell memory T cells. These cells become they migrate out of the lymphoid tissue and into the peripheral tissue where the effector cells are, or they develop into central memory T cells. Those central memory T cells can later also be activated and become resident memory T cells. Some of these, the majority of these central memory T cells will basically continue to divide and produce effector memory T cells. Those may divide a few more times, but not a huge amount, and lead to the effector T cell population, effector memory T cell populations. These are the effector memory cells. These are cells that have been reactivated and become effector cells. Once that infection is cleared, the fate of most of your T cells is apoptosis. So this model really kind of assumes a few things. It assumes that you've only got a small proportion of the offspring of an original naive T cell that commits to the memory lineage. That fits with the numbers of memory cells that we see circulating and in our bodies. The majority of the cells produced, like I said, about 90% immediately become effector T cell types. So this represents a pool of probably 10% or less of those cells that come out of that initial blast phase. But what they give rise to is a heterogeneous group of effector cells and memory cells that get generated early during the response. It's a big switch for me to think about them being made first instead of after. That said, there's still evidence that supports the idea that some memory cells do actually get made from the cells that survive the initial infection. Like T cells that are left over at the end of an immune response, some of them do acquire a resident memory cell phenotype or a central memory type.
that saying that the naive T cell has the least at least?
The naive T cell is completely undifferentiated other than CD4 or CD8.
Okay. I was for some reason I was thinking like, doesn't it have the most potential to?
It has the most potential to become any of those particular T cell lineages. So if it's a naive CD4 positive T cell, it can become either a memory cell or it can become any of the TH subsets that we talked about. TH1, TH2, TH17, T follicular helper. All of those.
So that just means like the level of differentiation that's happening.
That's just the level of differentiation, yeah.
When you said the lymphoid tissue, do you mean like both the peripheral and the central?
Central memory cells,
So central.
resident memory cells out in the peripheral tissues. Yeah. You need the CD4 positive T cells to be able to make memory cells. It requires actual help. We know that some of the cytokines, IL-7, IL-15, and Wnt is an intracellular signaling molecule, WNT. These play a role for making stem cell memory cells, but the pathway for this isn't totally clarified yet. We also know that IL-2 is important in generating effector memory T cells, which kind of makes sense. IL-2 was necessary for regenerating effector cells and getting that initial clonal blast going anyway. Another one of the major questions that kind of remain is whether the memory cells reflect the heterogeneity of effector cells from a primary response. So do we actually see all of those T helper subsets reflected in the memory cell pool? And the answer is unclear. The memory cell response appears to be diverse, but we've had trouble picking that apart too. People are still working on that. We're not sure if the diversity that we see is from the original response or whether it's generated fresh when some of those cells get reactivated. It could be either. It could be both. Differences between CD4 positive and CD8 positive memory T cells. We actually generate more memory CD8 positive T cells, so antiviral or intracellular pathogen T cells, than CD4 positive T cells, but they also proliferate more during their responses. There are also differences in the lifespan. CD4 positive memory cells don't last as long as CD8 positive memory cells. We don't know why, but it's interesting. We're not totally clear on how these memory cells live for as long as they do, because some of them can last for 70+ years. That is a very, very long ass time for a single cell to hang out in the body. You know, there's this myth going around the internet, and you know, you've probably heard it, that every 7 years you've got a totally new you. You've replaced all of the cells and tissues in your body. No, you have not. You have not grown a totally new brain in 7 years. Sorry. Your brain cells don't turn over that fast. Neither do your heart cells or your skeletal muscle cells. Your skin cells turn over faster than that. So it varies. Whether that's supposed to be some sort of an average is totally unclear, but even if it is, it's just not right. So that one, we can, just a little bit of logic will bust. And then we've got some of these memory T cells that persist for your whole lifetime. This is why we vaccinate young. The younger you can get the vaccines into kids, the more protected over the broader part of their life that they actually have that protection from that vaccine. Yes?
Which one proliferate more?
The CD8 positive T cells. It's just that during those initial clonal blast steps, after you activate a naive CD8 positive T cell, it actually divides faster and better than CD4 positive T cells. Like if you put them in a race head to head, CD8s would produce more offspring and do it faster. That may be evolutionarily necessary because virus replication is a very fast process and that can spread very quickly, whereas bacterial replication tends to take a little bit longer. We do know that there seems to be some dependence on particular cytokines to get those memory cells to occasionally divide and replace themselves. They will actually do that, including in peripheral tissues. If they're getting old and worn out, they can actually go through a round of mitosis and replace themselves before they completely, like, kaput. It's the technical term. What?
What does it mean?
Kaput, die.
Why do they keep dying?
Why do they keep dying? Because otherwise you would have lymphoma. Yeah. Lymphoma is a blood cancer that arises because of overproduction of lymphocytes. Those are sometimes T cell types and sometimes B cell types. So it depends on what type of lymphoma you're talking about. But yeah, overproduction of immune cells leads to too many white blood cells. That can actually cause problems too. Like they'll attack your tissues. They clog up your bloodstream. Okay. And we have reached the end of this chapter. Hooray! So this is where we stop for the next test. And also because I'm not going to try to start the next chapter with 5 minutes left in class. That would be stupid. So I will see you all on Tuesday if I don't see you at the review session, 6 p.m. on Saturday on Zoom. I will use the university scheduling tool thing like I did last time so that it's independent of my personal meeting room stuff and hopefully should not mess up.